So, welcome everybody to another Chatting with NDGS Paleo today on Wednesday, April 15th. Today we are going to be talking about bone histology and everything that we can learn about extinct animals by looking at not just the outside of the bone, which is what we do a lot to try to identify what animal we're looking at, but actually looking at the internal structure of the bone and what that tells us about say the individual age of the specimen. So we're not talking about how many millions of years old it is, but how old was that individual when it was alive up until the point where it died. Did it live for 20 years? Did it live for 15 years? And then also can look at rates of growth and things like that. So some really great information that we can get from the inside of bones. If you've been following along in our series for the past couple weeks, bone histology has come up a couple times very briefly. And so today, we're going to take a deeper dive into the science of bone histology. So leading us on that journey today is going to be Dr. Holly Woodward Ballard. She is an associate professor at Oklahoma State University, and she got her PhD from Montana State University doing, in part, this exact thing, bone histology, largely on a dinosaur called Maiasaura, which I'm sure she'll talk about today. So with that, I will hand off to Holly, and we can switch screen over to her, and she can lead us on our discussion today. Thanks for joining us, Holly.
Yeah, thank you. This is going to be fun. So I am a bone histologist, which means I study bone tissue microstructure, and in particular of dinosaurs, but you have to know a lot of comparative histology. So I study modern bone as well, because we have to understand what bone is telling us in modern animals to be able to make interpretations about the growth and the age of extinct animals like dinosaurs. So let's see, I have some images here that I can share with you guys. So I was just going to really quickly walk through the process of histology and sort of start to finish to give everyone an overview of what he is and how I go about starting from a bone like this to making a thin section I can actually see structure with under the microscope. So this is the proximal end of a Tyrannosaur tibia, and you know it's in one whole piece, but the first thing I have to do is cut a piece out if I want to look at it under the microscope. So I'll use a saw, in this case a tile saw, but it could be a band saw or something much smaller. The important thing to know is that it has a diamond blade on it so that it can cut through something as tough as rock, because dinosaurs are fossils have a lot of minerals in them, and so they're really tough. But after I cut a piece out, in this case this is the Tyrannosaur tibia, so it's hollow inside. Now I've got this great, awesome piece. Okay, so I've got a piece out. And then the cool thing about histology is that even though it's destructive, you can make a mold and cast of the piece that you've taken out, or you can nowadays even 3D scan. So that's what this image I'm showing here is of. It's not of the Tyrannosaur tibia. This is actually a Maiasaura humerus. And in this image on the right-hand side, you can see this little black bone sticking up vertically. That's the Maiasaura humerus piece that I'm scanning. And there's a little silver box sort of in the middle of the screen. That's my laser scanner. So it's very small and portable, but it gives you a completely three-dimensional digitized scan, which you can see on the computer monitor. And so before we actually cut the specimen up further or do anything else to it, we can make an exact replica of the specimen that we're about to make a thin section out of. So all the information is going to be there preserved. So you don't have to worry about, you know, it being destroyed forever, because we do have a replica of it. So after we have documented everything, photographed, scanned, mold, and cast, we can embed the specimens in a plastic resin. And this resin starts out as liquid. You pour it all over the bone, and then it hardens, and then we can make even thinner slices out of it. And the resin is there to stabilize the specimen, because if we just wanted to take a thin slice out, the fossil would just crumble into mud. So the resin is there to protect it as we take a smaller slice. And so that's what you're seeing in this image. There are several different bones here, and we've taken slices out. In this case, we've glued them already to a glass slide, but they're still pretty thick. They're a couple of millimeters thick, which isn't thin enough to have light pass through. So the next step is to polish them on a lapidary wheel, which is what this image is showing. And with the lapidary wheel, we use these silicon carbide plates. So they're basically like sandpaper. And you start out with really rough paper, and you keep going and changing the paper, making it finer and finer to get the scratches out and get the specimen thinner and thinner. And then eventually, as you're doing this, you can get it to where they're less than a millimeter thick. They're, in some cases, as thin as a human hair. And at this point, you can actually pass light through them and see them under a microscope. And the whole point of doing this is using a petrographic scope that passes light through the specimen. You can actually see internal bone microstructure. And that's the end goal, because now you can actually see what the bone record is telling you. And I'm going to stop the share for a second and bring up some really cool histology slides. Yay! Okay. So this is a really zoomed in image of dinosaur bone. And here's the scale bar. It's 0.2 millimeters in thickness. And what you're looking at here, all these beautiful colors, this is the dinosaur bone. And these the circles that you see in the middle of the colors, that's where blood vessels would have been in life. So most of the time, all we see are the hard tissue preservation in fossils. So we don't actually see the blood vessels, but we see the holes where they were going through the bone. And we can see the fossil bone, which is all this colorful stuff. And with the microscope, because I'm using polarized light, it's passing through the specimen in such a way that each color corresponds to the organization of a bone fiber. So all the greens, the fibers are all in the same direction. All the pinks, the fibers are in the same direction. So this can tell us about how randomly the bone fibers are organized, or how well organized they are, which gives us an idea about bone growth. So that's just a little quick overview. And I did want to bring up, if I can manage this, a little PowerPoint that will go through images and sort of what I'm getting out of all of this besides just pretty pictures. So this is just going to go over a little bit of what I've already discussed, but sort of in PowerPoint form here, we have this beautiful Maiasaura tibia, and within a bone, there are a bunch of different areas, and we're going to be focusing on the bone shaft, the middle part here, and we're going to be taking a slice out of the bone. And this is important because when we take the slice out, we can see different structures within, like I showed you with the Maiasaura histology here. What you're seeing are those holes where the blood vessels were passing through, and you're seeing the bone tissue organization around it. And what I was hoping to show you with this picture is that in life, bone is an actual living tissue. So you have cells that are forming the bone, and those cells are getting nourished by those blood vessels. There are nerves passing through bone. This is why it hurts so bad when you bang your shin or if you break a bone, because it is a living tissue and it changes. And this is why we can use that tissue to understand or interpret the record of growth for an animal. So we have our thin section slide. And again, with the microscope, this is what, in this case, a Maiasaura tibia looks like. in, in thin section, it looks a little pixelated because each one of those tiny little squares is one photo that this microscope and computer has stitched together. So in this case, the brown is bone and the white middle part is the marrow cavity. So it looks sort of like a ham bone or something like that. If you've ever seen a bone in cross section like that. But then when we zoom in, again, here is that bone tissue. The brown part is bone. In this case, the black areas are where the blood vessels would have been. And again, with that little schematic, we're looking at the top part here. So as I was saying earlier, with the Maiasaura tissue and the bone organization, the rate at which the bone fibers are deposited tells us something about how fast or slow bone was formed. So in something like this elk bone here, and again, all the pretty colors are the different bone fiber organizations, this elk was growing pretty quickly. And the bone tissue looks very similar to what we see in a large bird like a rhea. So a lot of people think, of course, that birds and mammals, they're not very closely related, but bone tissue is really cool because it can tell you more about how an animal was growing, and it doesn't really matter who's related to who. If you're growing fast, you're going to have a similar type of growth pattern. If you're going slowly, you'll have a similar growth pattern to other things that are growing slow. So this is really great news for us because we can use that in the fossil record and figure out how fast or slow unrelated species are growing, because it has to do with how the bone is formed. So slow growing bone looks more like this. So this is from an alligator, and you can see how much more well organized the tissue is. It's almost in layers, and you see that the blood vessels, which are here, these larger black circles, there aren't as many of them. Slowly growing bone doesn't necessarily need as much nourishment as fast growing bone. And because it's so layered, it tells us that this bone was being formed at a very slow rate. And because it's being formed slowly, it doesn't really need, the tissues aren't very disorganized. And like I said, it doesn't need much nourishment. But another cool thing that bones have, and probably the most important thing for paleohistologists, are these lines of arrested growth. And with lines of arrested growth, it's very easy to equate them to tree rings, because most everyone's seen a tree cut down, and you can look at the rings inside and you can count them and figure out how old that tree was when it got cut. The same thing is present in bone, and it's not just dinosaur bone. It's present in mammals, it's present in birds that take more than a year to grow up. So even humans and other primates have these lines of arrested growth. And by studying modern animals, we know that these lines are formed annually, and they represent a period of time in which growth stops. And this is different in different species, but for something like an alligator, it can be 3 to 4 months that they're not growing. For something like an elk, it's usually 3 months. So these lines just represent a period of time where growth has completely stopped. And in this image, we're looking at a partial femur cross-section from the Tyrannosaur Jane. Jane and Petey in this case are specimens from the Burpee Museum. They're both tyrannosaurs. And this is a close-up of Jane's femur, a little fragment of it. And these arrows each point to one of those lines of arrested growth. And the bone surface is to the bottom left. And all that brown, again, that's bone. All those little tiny circles you see are where the blood vessels would have been. And the arrows show you the spacing of lags. The cool thing, besides being able to count these lines of arrested growth, is that the spacing between the lags tells you how much growth the animal did from year to year. So you can see years in which Jane, in this case, was growing very quickly, the rings are spaced far apart. And the years where it wasn't growing very quickly, the rings are spaced more closely together. So again, like a tree and tree rings, you can tell good years and bad years from the spacing of growth rings. Another really cool thing about bone histology is that you can tell whether or not an animal was an adult when it died. And this is because we can find a structure called the external fundamental system. Histologists like to come up with these really, really long words and then abbreviate them down to like 3 letters. So this is an EFS, and in this case, these 2 images are showing you the bone surface of an alligator on the left and ostrich on the right. And you can see deeper in the bone, which is further down to the bottom of the screen, the tissue looks way different from that tissue that's right at the bone surface. And that's because the tissue at the bone surface in both of these cases were deposited very slowly. So you get a nice ordered arrangement of bone. You don't get many blood vessels going through it. And this is something we see in animals that have reached adult size today. So in this case, I'm showing an alligator and an ostrich, but we also see this in mammals. And we can find this in the fossil record as well. But when we don't find this EFS, in the case of the Tyrannosaurus Jane and Petey, we know that they're still growing. So Jane and Petey, these tyrannosaurs from the Burpee Museum, were important recently in a study that I was part of because they showed that these small tyrannosaurs were juveniles. They weren't pygmy adults because they don't have this EFS at their bone surface. So both of Jane on the left and Petey on the right were still growing when they died. So this gives us even more clues as to whether or not we're looking at juveniles or adult specimens in the fossil record. And that's really all I have as far as, a tutorial slide goes. And I can show you more images later. I can pause to ask questions, whatever you guys want.
Well, we can definitely dive through some of the questions first.
Sure, yeah.
And then if you have appropriate slides, you can always throw them up on the screen.
Yeah, that's what I thought. So I could just, stop droning on and actually answer some questions.
Somebody is asking if you've ever seen not just the cavity of where a blood vessel was in a bone, but an actual blood vessel.
I think that I have in some really well-preserved Tyrannosaurus bone. It looks like 3D preservation, so I don't just see the whole, but I see like this almost tree branch looking structure with little red balls in it that look almost like the heme component that Mary Schweitzer published on in some Tyrannosaur specimens. But so it's morphologically consistent. That's what we say when we haven't actually done the chemical analyses. So it's morphologically consistent with an actual blood vessel, but I would have to send the specimen to Mary Schweitzer or someone like that to actually confirm that that's what I'm seeing. But it does look pretty amazingly like a blood vessel.
We have the classic, what's your favorite dinosaur?
Oh, okay. Well, I have to cop out on that one and say that I have 2. T-Rex, just because it's amazing. It's my favorite carnivore. But because I've studied Maiasaura for so long, that's my favorite herbivore. Just because it has a very special place to me. You always have a love-hate relationship with your dissertation specimen. So that's my dissertation specimen, and it's still giving me cool information to this day. So I have 2. I can't just narrow it down.
And speaking of the Maiasaura, we have a couple people in chat who know who you are and are very interested in this critter. So if you can go into a little bit more detail on what you were doing with the Maiasaura, that would be amazing.
Yeah. So Maiasaura was named in the late 70s, and it's a really amazing specimen because it was actually discovered from baby bones. So the first specimens of Maiasaura were hatchling size, and it wasn't until the excavation really started that an adult skull of Maiasaura was found. And Maiasaura, it's a duck-billed dinosaur. It doesn't have sharp teeth. It doesn't even have an amazing crest on its head. But what's really great about it is that these little hatchlings were all found clustered together, and they were found within what looked like a nest. There was eggshell fragments everywhere, but the little hatchlings were too big to have just hatched out of the egg. And because they were still all together and there were other Maiasaura fossils around, it looked like this might have been a case of parental care. So they were staying in one spot. They weren't running around getting their own food. So this was not only the first baby dinosaurs ever found in North America, but it was also the first evidence ever of possible parental care in a dinosaur. And since then, we've found so many fossils of Maiasaura in the same area, ranging from those hatchling-sized animals all the way up to adults, and hundreds of individuals. And so this has given us a great opportunity to learn so much about one particular species. And in this case, we think that the hatchlings were helpless. They had cartilage pads on the ends of their bones. So that means that they probably couldn't get up and run around when they hatched. Karen Chen and others have studied Maiasaura coprolites, the fossil poop, and so we even know what it ate. It liked conifers and rotting wood, of all things. And through lots and lots of histology, we know that Maiasaura grew to its about 20-foot adult length in about 8 years, which is super fast. And let's see, what else? It didn't have a very good outlook when it hatched out. Most of Maiasaura died before they got to be 1 year old. But if they could reach 2 years of age, from about 2 to 4 years, they were pretty successful in growth and surviving. And then when they started to get to adult size, they were either getting ill or getting targeted for predation. And so the oldest Maiasaur specimen that I have histologically examined is 15. So we don't have anything older than that for Maiasaura, and that was pretty ancient for it. Also, Maiasaura was reproducing at 3 years of age, so it was reproducing before it actually reached adult size, which is pretty cool. So we know a whole bunch about it, and that's because of the amazing sample size. I have a graduate student right now continuing the work that Jack and I sort of developed for several years, Jack first, and then I picked it up, and now my graduate student is continuing looking at other bones in Maiasaura to try to understand more about its life.
We have a question on, if you can go over again, if mammals have the lines of arrested growth.
Yes, they do. So it looks as if any animal that takes more than a year to get to adult size has lines of arrested growth. Even people do. The difficult thing about finding lines of arrested growth in humans is that in addition to pausing growth annually, because your bones are getting bigger as you grow, you're changing bone shape. And when that bone shape changes, we lose some of that primary record. So human bones change shape and totally remodel every 7 to 10 years. So it's very difficult to find those rings or lines of arrested growth in humans because we're constantly remodeling our bone. So you can only really see them well in samples of young people. So by the time you're even my age, it's very difficult to find those rings in the bone. But other mammals do have them. They're pretty evident in something like an elk or a cow. So yeah, they're pretty prevalent in anything that takes more than a year to go up. It's not just a reptile thing.
I'm learning new stuff every day. I knew reptiles had very, very distinct ones. And I love how bone is just such a dynamic organ and that it's just constantly remodeling. It's just amazing.
Well, that's the amazing thing to me, is that it was, for the longest time, just assumed that reptiles had this and not anything else. And what really strikes me as interesting is how much we still don't know about modern bone. And most of that's because you can study animals that are alive today and we know how they're growing, and we can mark and recapture and watch it grow. So there's really no need to look at the bone tissue. But because of that, we don't really know much about bone growth and bone tissue compared to other ways of telling how things grow. So it's only really recently that we're starting to investigate bone tissue as it relates to growth.
We have a question here that we can answer. It's, has Maiasaura been found in North Dakota? No, it has not. And a secondary Maiasaura question, can you tell male and females apart from the bones?
Oh, that's a really great question. So far, we don't have any evidence that we can tell males and females apart. With Maiasaura, it's a huge sample set, so I expect that if we can find any kind of size differences, it would be pretty evident in the Maiasaura sample. And so far, we don't have evidence of that, but that's a great question because that's one thing that my graduate student is working on right now. He's going to be looking at not just the leg bones, the tibia of Maiasaura that I looked at, but he's also looking at the forelimb or the humerus of Maiasaura and looking at it histologically. And what we're hoping to do is, by looking at a variety of different bones in Maiasaura, see if there's any pattern, if there's any separation in sizes, that you get a bimodal distribution. So you get two size groupings that might tell us that there's a size difference between males and females. And he's also looking to see if we can find anything histologically different between the two. So that's to be determined, I guess, but it's a really great question, and it's very important, and it's something that one of my graduate students is working on right now.
How many dinosaurs have you cut up?
Oh, that's really hard. I haven't actually counted the number, but I've cut, the species I've cut includes Tyrannosaurus rex, Gorgosaurus, Albertosaurus, Triceratops, Hypsilophodonids, so the small little plant eaters that were bipedal and sort of a wastebasket taxon. I've cut several of those, and sauropods, of course. So Alamosaurus was the first type of dinosaur that I ever cut. I do have a wish list.
Yeah, I'm sure. And Clint has some questions.
Oh, yeah.
All right, Holly, I'm going to change this up a little bit.
Sure.
And I'm going to share my screen, and we are going to do an on-the-spot quiz show on bone histology.
Okay.
I'm going to show you, just to show our viewers how some of this works and how some of the interpretations work, and also some of the limitations of what we can tell from histology. I'm going to bring up some slides of dinosaur bones that Holly has not seen before, and we'll see what interpretations she can preliminarily make for us. Obviously,
okay.
these are very off the cuff, so we
no
won't
pressure.
completely hold you to this. So, but all right, can everybody see my screen?
Oh yeah.
All right. So I'll give you some background on these. So this image here, this is Teslasoros. It's a ulna, obviously transverse section through an ulna. So I can zoom into areas if you want me to, but just in general, you can just kind of talk through like what jumps out at you, what different types of stuff you see, what interpretations could be made from this, what is there not enough of.
Oh, sure. Okay. So this is cross-polarized light?
Yes, this is cross-polarized light.
Okay.
And it's a little thinner on this side, as you can see, which is why the color is a little off.
Yeah. So just, I like going sort of from low magnification to high. So
Okay.
this is where I would have started in the first place, and just looking at this in general. Um, on the left, where it is getting thinner, I see some radial bone, like right there where your cursor is. Um, that's something I see in areas of muscle attachment. So usually when muscle attaches to bone, it tugs on the bone, and the tendons inserting will cause the bone tissue in that area to look a little different from elsewhere. So that area in there looks like where a muscle would have attached. And it even has sort of a fibrous appearance to it where the tendon would have been inserting. So that's pretty cool. Um, Near the marrow cavity, so the center there, I see a lot of larger holes, which are called resorption cavities. And this is where bone is in the process of being remodeled or reabsorbed. And typically around the marrow cavity, you'll have these larger spaces. And then going out from that, I see just an amazing amount of blood vessel canals. So this is something we would call well vascularized tissue. And it's also very woven. So we talk about woven tissue as being disorganized bone fibers. It has a very, it almost looks like a basket weave pattern. And this is what we see in bones that are growing fairly quickly. So yeah, you see the little holes where the blood vessels are, and then around that is the quickly deposited bone fibers. And of course, probably what jumps out at everyone are the two rings that I see there. So those would be growth rings. And in this case, they're not solid, definite lines like we saw in Jane the Tyrannosaur. These are a little more fuzzy, but you can see how the tissue there is organized and bright. That means that it's going from quickly deposited bone, suddenly very slowly deposited bone, and then back to quick. And this is what we see in bone that pauses or slows down growth rather than pauses. So we don't have a definite stopping point in bone growth. Instead, it just slowed the growth down very dramatically for that period of the year and then sped up again. So instead of like a definite line, you have a diffuse line. So I see two of those. And then near the bone surface, it looks like, yeah, there we're getting more of that organized bone, which under polarized light looks very bright and shiny. And so this is telling me that it's possible this animal was reaching its adult size, because if you think about the way people grow, you just don't continue growing fast your whole life. You reach a point after your growth spurt that you slowly reach your final size. And that's what it looks like we're seeing here, that this animal had grown very quickly through several years and then was reaching its adult size because growth is slowing down. So these are the thoughts that go through my head whenever I'm looking at a bone, and hopefully I pass the test.
Well, this one's kind of a trick question, because this has been a weird one for me. So
Yeah.
So this is, like I said, this is the ulna. So for our viewers, the ulna is one of your lower arm bones. So between your elbow and your wrist, you've got two bones in there. One of those is the ulna. So this is a cross section. I said transverse, so it's kind of cutting right through. If someone was to chop your arm off, that's the section you would see. But to supplement this from the same individual, I have what's called a longitudinal section. So cutting it right down the length of the bone, starting at the elbow. So what we call the olecranon process. So that's the next image I'm going to show you.
Cool. Oh, neat.
And so that's the longitudinal. So this is the elbow up here going down, and this is about a quarter of the total length of the ulna. So it's the proximal quarter, the quarter closest to the elbow.
Mm-hmm.
So I don't
So
know
one,
how well this jumps out to you. I can point out what I've seen that's
sure,
strange,
yeah,
and you can give me your interpretation.
yeah.
So what I'm seeing is you've got this very this very open, porous, calcified structure through this part. But if you
Mm-hmm.
look towards the edges on this bone, you get, I can point you back, you can see this very clear differentiation right here between this bone looks, or this tissue looks very different over here from this tissue here. And you
Mm-hmm.
can see that that contact kind of comes down through here and kind of like that.
Mm-hmm.
And then on the opposite side kind of does the same thing.
Yeah.
Where it comes in like that. So to me, I'll get a little technical here, but I'll try to explain it. Almost looks like the formation of a bony collar.
Mm-hmm.
And that the inside here was still calcified cartilage or some very disorganized bone, and you've got more structured bones starting to form down towards the middle of the shaft, but the elbow joint at least was not fully ossified. So what are your thoughts on that, and how does combining that with the cross section, does that
Mm-hmm.
change your interpretation at all?
No, this makes sense because in dinosaurs and birds and other reptiles, they don't have a definite secondary center of ossification, so they don't have the bone or end caps that mammals do on their bones. They have what's called a cartilage cone. And so instead of the ends of their bones turning into mineralized, just hard ends when they stop growing, they stay fairly cartilaginous, and that's what you see towards the ends of the bone. And I think you're right that the stuff near the edge is the bony collar. And the cool thing about a bone when it grows is that the stuff that starts in the middle of the shaft, as you are elongating the bone, it ends up on the ends of the bone, because you're stretching the bone out as well as growing it in thickness. So what you're seeing there could be from it used to be more in the middle of the bone, and now it's being brought up towards the end of the bone, just because you're growing away from that midpoint. But what I think is really interesting about this piece is that a lot of times, especially in animals we see that aren't using their forelimbs to walk around on, that you see a lot of bone remodeling, and the ulna is usually one of the slower bones to form. So you usually see very slow growth with lots of bone remodeling, but that's not what I'm seeing in this. So that's really cool that it seems to be growing pretty rapidly for a forelimb, which they don't normally seem to grow as fast as the hind limbs do.
Yep. So this is, like I said, this is Teslasaurus. This individual is about 2/3 the size of the approximate full adult size that I've seen in Teslasaurus. Mm-hmm. But then again, you have to also contend with, you can have adults that reach different sizes. So that doesn't mean necessarily this was a younger individual, but it's been kind of interesting for me to see the differences in this this ulna compared to, I've got some from other Teslasaurus specimens that look very different, but not through the ulna like this, unfortunately. Mm-hmm. But that's what I was wondering, is if this would maybe tell us a bit about what these animals are maybe doing with their front limbs as well.
Yeah, that's a really interesting question, because not many people have looked at what the histology, biomechanical histology would tell you, whether or not, or how they're using their forelimbs. And that's another thing that my graduate student is working on with Maiasaura, to try to figure out what it was doing with its forelimbs. But that ring close to the surface on the ulna and transverse section, one thing about dinosaur bone and bone in general, if you don't have an entire complete cross section, like you're missing some pieces there.
Mm-hmm.
It's hard to tell whether or not, in this case, that ring at the surface is actually indicating that growth is slowing down, or it could have just died after its after a stressful period. So it stopped its growth and then it died. And maybe later the tissue would have resumed looking like it does everywhere else, which is why growth series is so important, so that you know whether or not you're looking at a lag that's at the surface or if you're actually looking at an EFS, because that can be pretty tricky. And it's really important for growth series in that way, just so you can be sure when you have an adult versus something that just died after a stressful season.
Okay. So like I mentioned for our viewers, this was a dinosaur called Vesselsaurus. Teslasoros is one of those, quote, hypsilophodontids that Holly was mentioning. So they're lower down on the evolutionary tree of the ornithischian or plant-eating dinosaurs. But this is one of the last dinosaurs to live. So it lives right to the end of the age of dinosaurs, about 66 million years ago. That's the end for me. I'll give the screen back now. So if you've had any other questions pop up while we were going through that, Becky.
So the first one is actually can be answered by both Clint and Holly. Why do we have the term wastebasket dinosaur, and why is a megalosaurid in the hypsilophodonts a wastebasket dinosaur?
Go ahead, Clint.
Yeah. So for the hypsilophodontids in particular, when the field of paleontology was first discovering dinosaur species, you had some very obviously unique and different species that were found, like your armored dinosaurs, like your stegosaurs and your ankylosaurs. You had your duck-billed dinosaurs like Maiasaura or Edmontosaurus. You had your ceratopsians like Triceratops that have very distinct features that all group them with each other more closely than anything else. There was also some relatively smaller dinosaurs that were being found that were most likely plant eaters or omnivores. They could eat both meat or plants, but they don't have these very over-the-top specializations that make it very clear what other dinosaurs they're related to. So what early paleontologists did was take all of these species and put them into a group that they called the Hypsilophodontidae, which was named after a dinosaur, one of the first dinosaurs discovered from England, called Hypsilophodon. So everything that was relatively small, so you're talking about like 2 to 5 or 6 feet long, that walked mostly on 2 legs and had very simple skulls with no horns or crests or anything, got lumped into this group called the Hypsilophodontids. And that's why we call it a wastebasket, because they mostly got put there because we didn't know what to do with them, because we didn't have enough information about dinosaurs and all the different species and how they were related to each other. Now, as our knowledge has moved forward, we've slowly been piecing out, oh wait, this guy that used to be over in the Hypsilophodontidae actually goes over with this group of dinosaurs. It's just a very early form. And this group goes over with this other group of early dinosaurs. It's just an early form that if you look really close, there are some subtle features that now we can tell pulls it over there, but before we couldn't tell that because we didn't have complete enough specimens. So over time, these wastebasket taxons get pulled apart, and the name kind of still sticks out there because so many people are used to that name, but eventually we figure out where these animals actually go. So for my PhD dissertation, part of what I did was work on these hypsilophodontids and figure out some of their relationships and move them to hopefully more accurate parts of the tree. There's still a lot that we have to learn about these animals, but we no longer think that they're all one group. We now know that they're spread out across the ornithischian or plant-eating evolutionary tree as the very early forms before they all became very distinct and different from each other. So when we use wastebasket taxon, what we mean is a bunch of species that we didn't know enough about to tell what they were actually related to, so we put them all together until we could fix it later. A similar thing happens in mammals, where you have a group that's as collectively referred to as insectivores, which is basically any small mammal that probably ate insects, that early on we didn't really know their relationship, so they all just got called insectivores. And now there's many, many different groups that have been pulled out from there and put on their correct places on the tree.
Okay, so we have I'm going to combine a couple of questions because they're relating to each other here. So talking about bone growth, some animals, like for instance, my leopard gecko, if she were to lose her tail, could regrow it back. How does that bone growth work, and why can't we grow limbs back? And what grows back first on the reptiles, the bones or the flesh?
Oh, those are good questions. So this is not my area of expertise, but from what I understand, the reason why we can't grow things back, whereas other animals like the gecko can, is because we're evolutionarily what was called more canalized. So we've lost essentially that ability through being more, going down a different evolutionary pathway. So we've essentially lost that ability to regrow a finger, for example, if it gets chopped off. But it looks like, from a paper I read recently on Permian reptiles that could regrow tails, that the bone starts first, but you've got, it's not like bones that's just sticking out there. So you're going to have the flesh that covers that little stub, but that it's essentially a part of the vertebra that breaks off and not the entire piece. So it sort of severs or planes in half and then can just get regenerated that way. I can tell you that I've read some papers that temnospondyls, which are really old vertebrates from back in the Permian, we have fossil evidence that some of them were in the process of regenerating limbs when they died. So I think that's really cool that we actually caught that in the act. But that's really the extent of my knowledge for that, unless Clint or someone else can help me out.
I do know that, like in the case of the leopard geckos, my leopard gecko is fine. She has a beautiful tail. We've taught my daughter, do not touch her tail, because I want it to stay nice and pretty. But we do know that on like the leopard geckos that lose their tail, they're not regrowing the individual vertebrae back again. They're just kind of growing back a substitute. And so any leopard gecko that has lost its tail does not get the nice, beautiful tail back again. It's just like this stub.
Yeah, it doesn't look as pretty. It's just sort of bare.
All right. The only other question that's popping out here, which is just kind of an amusing one, is, do bones get moldy?
I guess you mean like after death? So unless you have a problem, they shouldn't be moldy when you're alive. But fresh bones can get moldy, and I know this for a fact because if I want to study the skeleton of something freshly dead, and if it stays damp or isn't dried out completely, it can get moldy and fuzzy. Fossil-wise, a fossil can't really get moldy, but what you can find evidence of is bacterial invasion that totally screws up the bone histology record. So that mold that was on the bone as it was dying and decaying, if bacteria got inside the tissue and while that was happening before fossilization, it can totally screw up the tissue that you're looking for, and you won't be able to see it.
Yeah, let me jump in here quick again and share those images one more time. That's part of what we've thought. You can kind of see in these areas here on these Teslasoros specimens. We get areas where the bone histology is just completely obliterated. And in some specimens, you see a little bit through some of these, but we had a couple different individuals, it's mostly here on this one, but a couple different individuals, different specimens that we try to do bone histology, and just, even though the bone looks good when you cut it, the inside is just obliterated by something. And we don't know if that was, it sat out for long enough that bacteria or maybe a fungus got in there. But it seems like in some of these specimens, we just lucked out and the bone histology got destroyed, which is something I hadn't realized could happen before we did these sections. You can see in this area, it almost looks like something grew in and just fractured apart the bone through here. So it's another interesting thing to think about, and maybe tells us something about, you know, when this animal died, it must have either laid out on the surface or was very shallowly buried for a while, enough for this type of mold or fungus to grow and invade before it got deeply buried and preserved. So maybe tells us something about preservation, but it's interesting. I never expected to find something like that before. I don't know what your thoughts on that are, Holly.
Yeah, I've seen this before in several different fossils that I've looked at. And even when preparing modern bone slides for examination, alligator slides are notorious for that kind of thing, where you just have some kind of bacterial fungal invasion of the bone, and you just can't see structure. You get these black blotches on the slide where you can't see where the blood vessels were, you can't see the bone tissue. It's just black fuzz.
Okay, I think we've reached the end of our little Q&A session. So thank you so much for spending your morning chatting with us. I guess the only other request that I had is I saw that you had a folder of the really, really pretty stained glass pictures.
Yes.
Do you mind throwing up a couple of those before we call it quits?
I think, you know, I love bone tissue histology, but I think that teeth make some of the prettiest thin section slides. And this is a hadrosaur tooth. So a ductile dinosaur tooth that we're looking at in polarized light. And so all that zigzaggy pattern, that's wavy enamel. So that's the enamel covering on the tooth. And hadrosaur teeth are really interesting because their enamel has this alternating deposition, where under polarized light you can see layers to it, and it's just this gorgeous zigzaggy pattern. And below that, the brownish area that looks very fibrous, that's the dentine part of the tooth. And so all those fibers are tubules, basically going from the pulp cavity all the way up to the enamel. And this is what causes so much pain during a toothache. So all those little tubules are essentially carrying nerve bundles and nourishment through them, through the dentine. But it's, again, amazing to me that even something like that can be preserved for millions and millions of years. So that's a hadrosaur tooth. This is more dentine tubules, because I think they're just gorgeous. This is in the tooth of a raccoon. And this is polarized light with a wave plate. And the wave plate is the thing that makes polarized images just gorgeous, because it brings out purples and pinks. And again, all those little black wavy lines, those are your dentine tubules going through the dentine. And the tissue that they're going through is refracting light in different ways and making those gorgeous colors. So even modern bone has gorgeous tissue. You can make images out of that. This is that raccoon tooth again, but less magnified, so that you can actually see some of the structure. So the enamel is that little crescent there on the right. The pink is that wave plate. So the crescent on the right is the surface of the tooth. And that definite crescent line is the boundary between enamel on the right and the dentine and the tubules on the left. And again, I think this would make a gorgeous poster. And then this one is, we're back to bone again. So this is Tyrannosaur bone. I think this is Albertosaurus. And so what you're looking at are growth rings, so pretty definite rings, but then the tissue organization is different almost in between each one of those rings. So you get vascular patterns doing different things between each ring, and you get just these gorgeous color patterns between the bone, which basically is telling us that each year this dinosaur is growing a little bit differently, maybe in response to food abundance or shortage, but it leaves this just beautiful pattern in the bone. This is more Tyrannosaur bone. This is more typical of what you see in tyrannosaurs. This is I love it just for the uniformity. But all of the pink, that's bone tissue. Those black long squiggles are the blood vessels. And here are some of those more 3D looking vessels that I was talking about earlier. You can actually see the vertical ones cutting across the bone tissue. And again, they look more 3D to me than in other slides. But this pattern is called plexiform bone tissue. The vascularity has kind of a brick pattern to it. And this is very typical of theropod dinosaurs in general. And I think, this is the last one. This is remodeled tissue in a tyrannosaur bone. So as bone grows and changes shape, you get bone resorption. So bone basically gets dissolved away, but then bone can also get redeposited. And then you get these big circular, we call them osteons. So you get these circular osteons deposited back into the bone tissue. And it looks pretty disorganized and random, but it also leads to some really pretty colors. And again, all the little black circles are where blood vessels would have been. But these osteons typically reflect or refract light in different ways and give you just gorgeous pictures like this one. So that's my art show for dinosaur histology and modern bone histology, which I think is they're just amazing, beautiful things.
Very, very cool. Thank you.
Makes for good wallpaper on your computer.
Yeah, I could see that. Yeah, when I was running through my undergraduate years, we had to choose between different classes, and so we had the choice of genetics or histology. It's like, oh, histology, totally histology.
Mm-hmm.
It was a lot of fun.
So thank you, Holly, again for joining us. This was
welcome.
Excellent, and thanks for playing along on our Q&As and our little games.
Mm-hmm.
I hope everybody enjoyed that, and people should go back to some of our previous talks, like for example, the Tyrannosaur talk where we talk about bone histology, and with your new knowledge you've gained from this chat, you can go back and maybe understand what we were talking about there a little bit better. Moving forward, tomorrow we will be back again, 10 a.m. Central. We will be talking with Dr. Emily Bamforth. She is from the Royal Saskatchewan Museum, and she is going to be talking to us about plant fossils from the age of dinosaurs and how we can use plants then to interpret what the environment was like and reconstruct the world that the dinosaurs lived in. So Saskatchewan is, for anyone who's not aware, just to the north of us here in North Dakota, so it's very relevant for the environment here in North Dakota as well. So join us again, 10 a.m. Central, and one last time, thank you, Holly, for joining us, and we'll see everybody tomorrow.
Thanks for inviting me. It was fun.
Yeah.